Hi everyone, this is Dr. Santosh. Today I'll be talking about the nucleosome. Nucleosome means DNA plus histone. So in this lecture, in this video, I'll be talking first about the structure. What is the peculiarity of the structure of histone? That leads to a peculiar properties and then function. That properties lead to function. So I have divided this into these three objectives. So I'll first talk about the structural aspect of nucleosome, the very important part of nucleosome, that is the histone charge, the charge aspect of histone, then histone tail, and then DNA histone interaction. These are very important part of histone and nucleosome. Okay, then second part I'll talk about the dynamicity of that nucleosome. So nucleosome is not a rigid structure. How the nucleosome is dynamic, we will talk about that. And dynamicity of nucleosome is very much responsible for epigenetic modification. We will talk how that dynamicity is responsible for epigenetic modification. And then third thing we will talk about that this epigenetic modification, this modification in histone and nucleosome will lead to different phenomenon. That is bar body formation is because of that modification. Differentiation, you know, the one zygote, we were one zygote sometime, okay, in the beginning, but one zygote converted into whole body, different kinds of cells. This is act of differentiation. So how it is differentiated, it is all responsible, all credit is given to the nucleosome modification. Okay, dynamicity and modification of nucleosome. And then how this external environment, internal environment or hormone affect that nucleosome, we'll talk about that. So these all things we will talk about in this lectures. So let's start. You can see these are the nucleosome, okay, the DNA part here, you can see DNA part, and then these are the protein histone part at the center. So this is basically nucleosome. Any nucleosome is the protein that is wrapped by DNA. Now how this DNA and protein interact with each other, this is the very important question you need to know. Basically, you know, histone, if you see these histones, this histone protein is very rich in two amino acids. One is lysine, other is arginine. So you may wonder, because the name is histone, should be histidine amino acid more, but it's not like that. Lysine is around 20%, you will see, and this is also around roughly around 20%. So 20% you have the lysine. So lots of positive charge. These are lysine and arginine. These are positively charged amino acid. Okay, positively charged amino acid. So you can see there are lots of positively charged amino acid here, residue here. Okay, the side chain of these amino acids are positively charged. These are because these are basic. So lots of positive charges are there in the histone. And histone is basically 8 protein joined together. This is octamer. Histone protein is octamer. And you know, this protein has lots of positive charge derived from lysine and arginine. Now this DNA, if you see the DNA is backbone of DNA is, if you see, nucleotide phosphate, other nucleotide phosphate, okay. So this nucleotide phosphate is there in the backbone. This phosphate is negatively charged in DNA. That backbone, if you see, this backbone is made of negative charge because it is phosphodiester bond that makes the backbone of DNA. Here the backbone of DNA is made by phosphodiester bond. Now this phosphodiester bond, negatively charged DNA, now when it come in contact with this histone, which is positively charged everywhere, now it will easily wrap around the histone. So this is the main thing, main concept, how this DNA interact with histone. DNA interact with the histone with its negative charge. Histone has its positive charge. So negative charge of DNA and positive charge of histone will combine with each other, attract each other, and then form this histone octamer. Okay. So this is how histone octamer is formed. Now, and you can see this is histone. Basically, if you see the arrangement generally in DNA, so this the DNA will wrap the histone. Okay, the DNA segment, if you see the DNA segment, the nucleotide of almost about 140 7 base pair will be there. You can remember roughly 150 base pair will be wrapping around this DNA, almost twofold. Okay, 1 plus 3/4 fold, like that. So it's almost two times it will wrap around the histone. Now, and there will be in between, there will be some distance, some linker DNA, and then here again. So this, what is the purpose of this histone folding? The main purpose of this DNA folding over the histone is to protect DNA. Okay, protect DNA. Why DNA need to protect it? Because DNA is not a stable molecule when it is open like this. When it is open like this, it is not a very stable molecule. It can be degraded easily, and it can be, the injury can happen easily. So DNA has to be preserved. And how to preserve? By losing some of the energy by folding around the protein. So when it folds around the protein, it is protected. Now, so it is protected. That is one advantage. But what happens is, because of this advantage, you have a disadvantage. What is that disadvantage? Disadvantage is that because DNA has a code, many code, and that code has to be translated into mRNA. So this DNA, when it is in folded condition like this or like this, so the DNA transcription will have a problem. So there will be problem in transcription. So transcription cannot be done when DNA is folded like over the protein. Or if DNA, this will fold like when DNA protein will fold and compact more, then it will also be difficult for the RNA polymerase to approach a particular region. So these are the reasons. So when DNA is compacted over the protein, the DNA is protected from damage, but side effect is it is not approachable by transcriptional machinery. So now here is the solution. The solution to this problem is dynamism of this structure. So you have to understand, you see this structure like folded structure of this histone protein here, also histone protein on DNA, folded DNA over histone. It's not a rigid structure. That is a very important concept you have to understand. It's not a rigid structure. It is a dynamic structure. And to make this structure dynamic, it requires some of the protein, participation of some of the protein. These proteins are grouped into 3 categories. You can see this protein are called nucleosome remodeler. As the name suggests, nucleosome is DNA, and the protein, histone protein remodeler. The second is histone modifier, and third is histone chaperones. The first and the most important of this is this histone modifier, because this is the first protein, first machinery that approach this histone and this nucleosome, and then it will modify histone. We'll talk about this in very great detail. This is the most important part. Okay, what it does is it goes, and there is, you can see the tail part, it will go and modify the tail part, and that is how it will neutralize the charge on the histone, and then somehow this DNA will try to unwrap, okay, dislodge from the histone protein. This is how it happens in the beginning, and then next this histone remodeler will come and actually make the space for RNA polymerase to sit on the DNA. So this is how it happens. And histone chaperone is very important during DNA synthesis. What histone chaperone does is it completely takes out this histone from the DNA and put histone back into the DNA. So this kind of protein. So what this histone remodeler does is, this is the job of histone. It will push this nucleosome away from each other and creates gap here, and on that gap you can see several protein can come and sit, especially DNA polymerase and transcription factor will come and sit here, and then transcription starts, and further job of this histone remodeler will be there. Okay, so this is how histone modifier and histone remodeler works together. We'll first talk about this. We will mainly talk about histone modifier. To understand this histone modifier, this is this picture. You can see this is the figure I have taken. This is to show during replication how histone chaperone works. It will take out all the histone, okay, and it can put the histone back during the replication process, because during replication process you can see that there are two DNA. One DNA requires addition of more histone protein. So this is done by histone chapter. So we have done this three histone modifier protein that gives the dynamicity of the histone, this nucleosomes. So that gives a dynamicity to the nucleosome. So what happens is the part of DNA which is not required for transcriptional machinery, which is not transcribed, would be compacted and secured, made it safe, okay, from damage. Now the part which is required is only unfolded by this histone modifier and will be approachable by the transcriptional machinery. So let's talk about this histone modifier. Before talking about this histone modifier, let's talk about the histone structure in little more detail. Then we will understand how this histone modifier modifies histone. Let's see this. So you can say these are the this is the structure of histone. You can see this is the structure of histone. If you see the structure wise, it is octamer. It is octamer. 8 histone proteins are grouped together. Actually, it is a tetramer of this 2 tetramer of this 4 histone protein. So if you see, it is H2A, it is H2B, and then H3 and H4. These 4 type of histone protein, in 2 in number, they will form octamer. We don't need to know anything in detail. Just remember this is octamer. And here what we really need to understand is this tail. You can see this tail. This tail comes from this N-terminal part of this all protein. Okay, this tails are very important. Tail does not mean carboxy, this last end. It's the N part, the amino acid starting sequence are the tail. So these tails are very, very important. And why is it very important? I'll talk about this. So when you can see these are the histone protein, they will assemble and will form octamer. No need to know in detail. Okay, this is dimer. This is how they will assemble and form dimer. S3, S4 will form again tetramer, and this 2 dimer of this H2A, H2B, and 1 tetramer of this S3, S4 will come together and form octamer. Okay, now very important thing is the tail. Okay, now let's see this tail. Now you can see here in this picture, the tail can be modified. Tail can be modified by adding some group. Now these tails have a amino acid residue. Okay, let's say this is tail, these are the tail. This tail can have this amino acid lysine. Lysine has this NH2 group like this. Now it can have somewhere arginine. Arginine has this guanidino group. Okay, this type of group where nitrogen 3 nitrogen will be there. Okay, so this nitrogen can be methylated. Okay, can be acetylated. Okay, so acetylation or methylation, based on which group is added here, the histone can be modified. Now what is the effect of that modification? We'll see in a while. Let's see this. So you can see here K means lysine. So their sequence and DNA structure is conserved, very, very conserved along the evolution. Whatever DNA protein sequence is there in human, the same type of DNA sequence you will find in the eukaryotic single cellular organism. So here you can see this amino acid sequence are pretty much same everywhere. So now you can see here the most important, there are sequence shown, the most important one I want to focus here is S3. The S3 sequence. Other have this, but once you understand one very well, then you can apply the same concept in other. So this histone H3, okay, and 9th position means lysine, and 9th position here have lysine, so we call it K9. So 9th position of this histone H3 has lysine, and lysine is a terminal NH2 group. Okay, now this lysine can be methylated. You can see this red is shown, it can be methylated or acetylated. Now I'll tell you here. Now when any DNA protein, when it is acetylated, what is acetyl group means? Acetyl means negative charge. So negative charge is there, and DNA also has negative charge, so there will be repulsum. So what is that? Loosening of, when DNA is there like this wrapping the histone protein, when you insert in tail negative charge, so there will be repulsion in the nearby, you can see the DNA. So there will be loosening of this nuclear protein complex. Okay, there will be loosening of this nuclear protein complex. So this will create euchromatin, means switching on the gene. So acetylation switch on the gene. That is how acetylation switch on the gene. Now when methyl group is added, okay, when methyl group is added, charge is neutralized. The positive charge there that was holding DNA before is neutralized. Now what will happen as a result? When the charge is neutralized, there will be more compaction. So DNA will be more compact as a result of addition of methyl group. So remember, this is a general rule about methyl group. Okay, when methyl group is added, that methyl group will cause heterochromatin formation. And then when acetyl group is added, it will cause euchromatin formation. That means loosening of this whole structure, DNA protein structure. And now when it is loosened, so it is approachable to transcriptional machinery. So this heterochromatization means switching off the gene. So this heterochromatization, it will switch off the gene. This is switch on the gene. So acetylation will switch on the gene, methylation will switch off the gene. So let's see this here. Now you can see here histone has been, when histone is methylated here, now you can see the compaction of this whole protein. Now this histone, this all came together and compacted. This is basically heterochromatin formation, and this is called switching off the gene. So gene will be switched off this way. Now this DNA over here, it cannot be approached by any transcriptional factor or RNA polymerase. It cannot be approached by transcriptional machine. While if you see here, when you add on tail acetyl group, so acetyl is negative charge, so there will be repulsion, and then this structure will be loosened. When this structure is loosened, what will happen is, now the DNA polymerase transcription factor, they will get site, they will access, and then what happen, this will again hire nucleosome modifier protein, and then this nucleosome modifier protein will push this nucleosome far away to each other, and then it will create space. This is how, you know, the transcription machinery will work in eukaryotic organism. Just remember, prokaryotic does not have this problem. Prokaryotic does not have nucleosome. So prokaryotic DNA does not require this nucleosome modifier, histone modifier. So here we have histone modifier. I will show you there are many group of protein in our cell, and those protein, they will do histone acetyl transferase. We call them HAT enzyme. Very important to remember. You don't have to remember this all. I just, for a time being, I only want you to remember this. Okay, this is CBP P300, CBP 300. Other, I don't want you to remember for this video and for other time also. So this all enzymes, they are called HAT. Okay, histone acetyl transferase. What is the job of this HAT? Job of this HAT is to switch on the gene to form euchromatin. Okay, now there are a group of enzymes that will remove histone. So remove acetyl group. Addition of acetyl means on, the opposite will be off. So this all, job of this all will be off. So you don't have to remember this name, only I want you to remember this name. Now there will be a group of protein, okay, that will cause methyl transferase. Again, this will cause off, especially when methyl is transferred to S3, K9 position, it will be switched off. Okay, now when some gene, they will remove the methyl group, means it will cause switch off. So remember, you can see the methylation will cause switch off, and then heterochromatin formation and gene switch off, and acetylation will go switch on. This is very important to remember. Now this is HAT enzyme. This is what is called histone deacetyl. In short, we call HDAC. Okay, acetyl transferase, HDAT. Now this is histone methyl HMT, histone methyl transferase. Now HDMT. Okay, histone demethylase. HDN. So histone DNA. These are the enzymes that are required to modify the histone. The most important concept is histone. This nucleosome is a dynamic structure, and it can be modified by a group of protein. And the most important modification, and from which is actually beginning of the modification, is histone modification, then nucleosome modification. Okay, there are many proteins that were involved in histone modification. Okay, these are the proteins these are the enzymes that are involved in histone modification. Now let's see the consequence of histone modification. What are the factors that influences histone modification and a real application of this modification of histone? Let's see. You can see in like in female cell, if you see, there will be one bar body formation. This bar body formation is because of this methylation of this lysine at 27th position in histone 3. So let me take you to that position. You can see here, this is the lysine 27th position. Okay, and that will be methylated, and methylation of this lysine at 27th position in histone. Okay, in histone, let's say these are the histones. There will be histones somewhere. Okay, so this histones, when it is methylated somewhere, this is acetylated, so it's okay, it's open. Now when it is methylated, so it will clump together and it will become like this. So remember, histone methylation is the end consequence of this bar body formation, and this because of histone modification, bar body is formed. What type of histone modification? The H3K27 trimethylation of this H3K20 lysine at 27th position in H3 histone. Trimethylation of that lysine will cause formation of this bar body. So you can see this bar body inside cell in electron microscopy. Okay, you can see this bar body formation in electron microscopy here. Okay, you can see. So this bar body, when it is heterochromatin formation, almost all genes are switched off except few genes are switched on there. It is switched on. Almost all genes are switched off, and this switched on heterochromatin, they are always pushed to periphery. Anything we don't require, what we do? We put that into shell. The same thing cell does. Okay, anything cell does not require, it will compact and push into the periphery. That is what happens with the bar body. So this is bar body formation is also because of histone modification, heavily methylation of the histone. Which histone? H3 histone at 27th lysine which is present at 27th position is trimethylated. Okay, now let's see the other effect. You can see various growth factors, neurotransmitter, various growth factors. They somehow go and actually phosphorylate a protein that is bound to DNA. Okay, always this is CAMP, CRE means CAMP, can read from here, CAMP response element. Now this to the CAMP response element, there is always CREB is bound, that is CRE binding protein is there. Now these all factors will phosphorylate this. When it is phosphorylated, somehow higher some protein that will modify histone. This protein I told you, do you remember that protein? CBP. And this, what is the CBP? This CREB binding protein. This protein is actually nothing but HAT, histone acetyl transferase. What it does? It will transfer that acetyl group to the histone nearby to the DNA. Okay, when acetyl group is transferred, now this histone, the DNA will be loosened, and then transcription factor, okay, transcription factor and RNA polymerase will come and bind here, and transcription will happen. So this is how transcription is induced. So because of this nucleosome modification from external factor, this transcription is induced here. So this is how nucleosome modification is brought about, but different growth factors and other things inside the cell. Not only growth factors, there are other factors. Okay, for example, environmental factor that can also bring about that nucleosome. You can see this is just a model. You can see when cold is placed, so this will again higher this CBP, and there will be a loosening of DNA because of acetylation and then transcription. The same thing you will see like that. When environmental factor also can influence this recruitment of histone modifier, and histone will be modified. A certain gene will be switched on and another gene will be switched off. This is how environment will affect your gene, control your gene. Now these are the things you will see in, that one zygote, we all wear one zygote at some time, and that zygote has transformed into whole body. Now what happened during zygote formation? All cells were the same, but some cell they differentiated into the neurons, some cell differentiated into muscles, some cell differentiated into bone, liver, kidney, all other cells was derived from this. And this, how do they derive? Actually, if you see genetically, all these cells have the same gene, okay? The same gene, same DNA sequence. Only difference, you will see the set of gene. Let's say set 1 is active here, and set 2 is active here, set 3 is active here. Okay, the combination of, let's see, 1, 2, 3, 4, 5. If you see 1, 2, 3, 4, 5 genes are there. We have 20,000 genes. So just I'm taking a schematic. So 3, 4, 5 genes are active here. Okay, let's say this 1, 2, 3 genes are active here in the muscle. This 4, 5 are inactive. Now if you talk about this bone, let's say 2, 3, 4 genes are active here. Now third set is active here. Gene 1, 4, 5, they are inactive. This is how not all genes are active. Few genes are only active in a particular tissue. That gene will be required. As a rule, I told you this nucleosome is a mechanism to protect DNA. Only DNA that are required for that particular cell will be, will be in the loose form or switch on form or euchromatin form, and will be transcribed. Those which are not required will be pushed into heterochromatin form. So you will see insulin gene will be inactive form in all of this. Okay, it will be active form in heterochromatin form in beta cell of pancreas. So this is the thing you will see. Because of this nucleosome modification, you are getting different type of cell in your body because of this nucleosome modification. What influences nucleosome? Here, during embryogenesis, various growth factors are there, and environmental nearby cell influences, environmental factors of cell. So this growth and environmental factors plays very important role in histone modification that I have shown you. The bar body formation, I also told you that has influence on this. The histone modification plays a role in bar body formation. Now metabolic shift. When you keep eating, insulin is high. Okay, so what will happen? The insulin will induce certain gene that will be very important for fatty acid synthesis and fat synthesis, and you will slowly get fat. You lose weight, or then what you do? Glucagon, increase the glucagon level in your body. Okay, glucagon will switch on the gene that are responsible for fatty acid breakdown, and you lose the weight. And you'll become slim. Now tumor formation, let's say, because generally when it is switched off, your tumor gene, which is mutated gene, is switched off. Somehow you because of some environmental factor, you switch on that gene that has mutation in it, and that can cause tumor. So tumor development can happen because of switching on a bad gene. Okay, so this is all about this nucleosome modification. Remember, the last point is nucleosome is a very dynamic structure. Okay, but the purpose of nucleosome is to protect the DNA, because eukaryotes have lots of DNA, and the side effect is, when DNA is protected, the transcription machinery will not get access to those DNA. So here come the function of nucleosome modifier, histone modification enzymes. So histone modification is the most important part of all this dynamicity. Histone modification is the most important part of the protein that are involved in dynamicity of this gene. Thank you. Take care. I hope you understood this whole class. Okay, if you have any question, you can put the comment below.